go chronologically on this. So sampling all starts in the field. You can't, no matter what tools you have, you can't do anything unless you've got some sort of material or data to work with.
And so you're in rivers.
So I work in rivers. I'm pretty exclusively a river biologist. And so what I do is I go out with a team and a bunch of dry ice and a bunch of syringes, and we find animals that look like they're in trouble, and we draw a little bit of hemolymph, which is their equivalent to blood, and then we immediately snap freeze that on dry ice, and then we send it off to the labs, drive it straight to an airport, put it on an overnight flight, and within 12 hours of when I put it in the box and ship it off, it's here in a freezer in the lab, which is where he takes over.
Yeah, and what Jordan just described is a massive effort. So he has a fantastic team that works with him in his office, and they have, the amount of planning that goes into this is impressive, and the amount of spontaneity, because it's dependent on river conditions. So you can have the rain blow out the river, and then you have to just make a choice and go as soon as it gets to the right level. So keeping that in mind, we're very careful with what we do here, because it takes an enormous amount of effort. And we are using really some of the most advanced DNA sequencing technologies that have been developed for things like personalized medicine and the Human Genome Project to apply to this new system. So we will take those samples that Jordan sends and we will process them in a way that prepares them for random shotgun sequencing of all of the DNA and RNA that's in them. And we essentially create millions of DNA sequences at random and feed them into the computer. And the computer helps tell us what sequences are in there that might be from a virus. And then there's confirmatory testing and lots of other things, but basically that's how we first identify the viruses that might be in this system. And we can then explore those later on by looking at more animals or developing specific tests.
Both ends of this are really involved and take a lot of work, but they're both completely useless without each other. Without some kind of sample collected with the right level of care, preservation, attention to detail, and from the right places too. You can't just go out and grab hemolymph from wherever. Because we're using such unexplored animals, we have to pin all of our hopes on identifying the ones that are in trouble and then compare those to ones that are doing just fine. It's the case control model. So that's more associated with Tony and his group, and so I've had to sort of learn some of that and on the fly adjust in the field to get those samples, because without getting those right, we can't learn anything from them when they get down here.
So it's actually a really good point. There's highly involved field work and highly involved lab work, but the link is epidemiology. What we're trying to do is, albeit with fancy methods, just what epidemiologists have done for centuries, which is to try to identify the causes of health-related outcomes. So the first thing you do is you choose very carefully which groups or populations to compare, and then you come up with differences between them to arrive at associations. And once you have these associations, you explore them more fully to see if they might be causal associations. So what Jordan and I are doing is working together to get the appropriate population comparisons to find viruses that, or other microbes that might be associated with sick mussels. And then we have another group of collaborators at the U.S. Geologic Survey National Wildlife Health Center and the United States Fish and Wildlife Service and Geologic Survey Fish Health Center in La Crosse, Wisconsin, who are going to be doing additional tests, like making specific assays for these agents and even looking for where they are in the tissues of diseased animals. So we can go that extra step and say not just what might be associated with these declines, but might actually be causing them.